From clinic halls to late night rounds, questions in your mind, cases, clues, and patient care, all in one straight line. This is the Clinical Medicine Podcast, where practice and the evidence meet. From bedside chat to breakthrough data, clear, concise, and right at your seat. The Clinical Medicine Podcast, making complex things feel clear. Tune in, learn on, stay curious. We're glad you're here. Oh yeah.
Hey everyone, welcome back. So let's jump right into the amazing world of renal anatomy. Where are these kidneys actually located?
They are retroperitoneal, meaning they're behind the peritoneum, which is the sac that holds a lot of your abdominal organs.
Behind it, so they're kind of tucked away then.
Exactly, and they're wrapped in a capsule and a nice layer of adipose tissue.
Adipose tissue? Like fat?
Yes, fat. It's super important because it holds them in place.
Wait, so if you lose too much fat...
Then the kidneys can actually drop. It's called renal ptosis. It's wild.
No way. They can just fall down?
Well, not fall out, but they can descend. And in very thin people, it can cause the ureter to kink.
That sounds painful. So they're fixed in place, but they still move?
Oh, yeah. They actually move with your breathing, up and down a bit.
That's incredible. And what's their normal size?
Normally about 12 to 13 centimeters, roughly the size of your fist.
So if they're much bigger or smaller than that, it's a red flag.
Precisely. Size variation is a key indicator for many kidney diseases.
Like what kind of diseases?
Hydronephrosis or polycystic kidneys can make them huge, up to 25 centimeters.
Wow, you could actually feel that.
You absolutely could. It's a critical diagnostic clue.
So we've talked about how they sit in their normal size, but what happens if they're not that perfect 12 to 13 centimeters?
That's where things get really interesting clinically.
Why? What does size tell us?
Kidney size is a huge diagnostic clue. It's like a neon sign for what's going on inside.
So bigger is badder?
Not always. If they're really large, say up to 25 centimeters, we might be looking at something like hydronephrosis.
Which is?
Basically a swollen kidney due to urine backup, or polycystic kidney disease where cysts make them enormous.
Oh, so a doctor could actually feel those during an exam.
Absolutely. Those big ones can sometimes be palpable during an abdominal check.
That's wild. What about if they're too small?
Smaller kidneys, especially if they're shrinking, often point to chronic kidney disease.
Like end stage?
Yes, advanced renal failure. The tissue atrophies and the kidneys just get smaller and smaller.
So just measuring them gives you so much information.
It's a critical first step. An ultrasound or other imaging can immediately flag issues based on size alone.
It's like they're telling us their life story through their dimensions.
Precisely. The size variation is a clear pathological signal.
Okay, so we know how big they are and why size matters, but what's going on inside this incredible organ?
Great question. The kidney has two main layers, like an onion.
An onion, I like that.
The outer part is the cortex, the cortical layer. That's where you find the glomeruli.
Glomeruli, what are those?
They're tiny filtering units where the magic of forming pre-urine happens. It's crucial.
So the filtering starts there. And the inner layer?
That's the medulla. It's deeper and mostly has tubules and these cool structures called vasa recta.
Vasa recta.
Yeah, big collecting vessels after the initial filtration. No glomeruli here.
Just tubules and these big vessels. Fascinating.
And these tubules group together to form what we call renal pyramids.
Pyramids? Like the ones in Egypt?
Ha ha! Conceptually similar in shape, but microscopic. These pyramids end in structures called papillae.
Papillae.
Exactly. Multiple papillae then merge to form calyces, which are like little cups.
Cups. So things are getting collected here.
Right. These calyces eventually drain into the renal pelvis, which is kind of the kidney's funnel to the ureter.
So it's cortex, then medulla, then pyramids, then papillae, then calyces, then the pelvis. It's a whole system.
a beautifully organized system all designed for efficient filtration and waste removal.
So if those are the kidney's internal structures, what's actually doing all the heavy lifting for filtration?
That's the nephron. It's the functional unit, the tiny workhorse of the kidney.
The nephron. How many of those are we talking about?
About 1 million in each kidney. They're super small, but incredibly complex.
Wow, 1 million. So what are its main parts?
It starts with the glomerulus. That's the filtration part.
Okay, the glomerulus for filtering.
Then comes a long, winding tube, the proximal convoluted tubule.
Proximal convoluted tubule. Got it.
Next is the loop of Henle, which dips down, and then the distal convoluted tubule.
So glomerulus, 2 tubules, and a loop, and then?
Finally, the collecting duct, which gathers everything up.
So the glomerulus filters, but what do the rest of the parts do?
The tubules and the loop handle reabsorption, bringing good stuff back into the blood, and secretion, getting rid of waste.
So it's not just filtering, but also recycling and fine-tuning.
Exactly. That's the amazing part of the nephron.
It's like a tiny, super efficient water treatment plant for our bodies.
A perfect analogy. It's constantly working to maintain our fluid balance and filter out toxins.
So that covers the nephron, but what about kidney pain? Like when people say, my kidneys hurt.
That's a big one. Here's the wild part. The kidney itself, the organ, has very limited sensory innervation.
Wait, really? So it doesn't actually feel pain?
Not in the way you might think. That typical back pain people call kidney pain is often muscular or spinal.
No way. So my back hurting isn't my kidneys screaming for help?
Exactly. The actual kidney tissue doesn't have those nerve endings to signal pain directly.
Okay, so what parts of the kidney system actually do hurt then?
Well, the renal capsules surrounding the kidney and the urinary tracts, like the ureters and bladder, those are innervated.
Ah, so it's the covering, not the organ itself. Makes sense.
And when we talk about true kidney pain clinically, we're often looking at uroteral colic.
Uroteral colic, what's that?
It's this intense wave-like pain. It happens when there's an obstruction, like a kidney stone in the ureter.
Oh, like it's trying to push the stone out.
Precisely. The ureter goes into spasm, contracting hard to get past the blockage.
And that's what causes that sharp, terrible pain people describe?
Yes, that's the mechanism. It's the muscular contractions of the ureter trying to overcome the obstruction.
So it's not the kidney feeling pain, it's the tube connected to it.
Spot on. It's a crucial distinction for diagnosis.
So let's kick things off with the kidney itself. It's super important, right? But also really vulnerable.
Absolutely. Think of the kidney as one of the body's VIP organs.
A VIP organ? Like what, the brain and liver?
Exactly. It's right up there with them in terms of how much blood flow it needs.
Wow, so it's constantly working filtering stuff.
Precisely. It's highly perfused, meaning it gets a ton of blood flow, which is crucial for all that filtering.
Okay, so high blood flow equals high importance. Got it. But where does the vulnerability come in?
That very same high blood flow makes it incredibly sensitive to any interruption.
Interruption, you mean like if blood stops flowing?
Yes, exactly. That's what we call ischemia, lack of blood flow.
And that's bad, obviously.
Extremely bad. Even short periods of ischemia can lead to serious damage like tubular necrosis.
Tubular necrosis? What's that?
It's when the cells in the kidney's filtering tubes start to die because they're starved of oxygen and nutrients.
Oh, that sounds awful.
It is. The kidney is so delicate that ischemic damage can be comparable to what happens in the brain.
Wait, really? Like brain damage?
Yup. We're talking minutes, maybe just 3 minutes of interrupted blood flow can cause significant issues.
3 minutes? That's barely any time at all.
It highlights just how vital that constant, strong blood supply is for kidney function and survival.
So the kidney is super vulnerable, right? What about its plumbing, the renal artery?
Exactly. The renal artery is fascinating. It's the main highway in.
Okay, and then it branches out?
It does, typically into 2 main trunks, then those split into interlobar arteries.
Interlobar, like between the lobes?
Precisely. They run from the medulla, deep inside, right up to the cortex.
And then what happens?
They curve. They become arcuate arteries running parallel to the kidney surface.
Arcuate, like an arch. That makes sense.
Right? And from those, you get the interlobular arteries heading back towards the surface.
So it's like a whole network bringing blood everywhere.
A super intricate one. And these interlobular arteries are key
Why is not?
because they're what feed the glomeruli, where the actual filtering begins.
Oh, so the blood comes in through the interlobular artery, and then where does it go?
It enters a tiny vessel called the afferent arteriole.
Afferent arteriole, got it. And that leads to the glomerulus.
Yes, directly into the glomerulus. It's like the gatekeeper.
So the blood flow is super specific then to make sure filtration happens.
Absolutely. It's all about getting that blood to the filtering units efficiently.
And what about blood pressure? Does that play a role here?
Huge role. Adequate blood pressure is essential for proper renal perfusion.
So if your blood pressure drops, the kidneys are in trouble?
Exactly. Low pressure means reduced filtration and potential damage.
So we've talked about how blood gets there. What actually happens inside the glomerulus, that little filtering powerhouse?
The glomerulus. Think of it like a super fine coffee filter.
A coffee filter.
Exactly. It's this tiny, intricate network of capillaries all coiled up.
And that's where the actual filtering happens?
Yes, blood enters this network through something called the afferent arteriole.
Afferent, got it. It's bringing the blood in.
Right, and here's the cool part. That afferent arteriole has a larger diameter.
So more blood can rush in?
Precisely. It's like turning up the water pressure in a hose.
And after it's filtered?
The blood leaves through the efferent arteriole, which is actually narrower.
Wait, narrower? Why would it be narrower?
That difference in diameter creates pressure inside the glomerulus.
Ah, to push the fluid out.
You got it. It forces water, electrolytes, and glucose out of the blood and into Bowman's capsule.
So that's the pre-urine that we're talking
The
about?
very same. Now, here's what's super important about that filter, that membrane.
What about it?
It's designed to keep big things like proteins and blood cells from getting through.
So no protein in the pee, ideally.
Exactly. If proteins start showing up, it's a red flag.
A sign the filter's broken?
It's a strong indicator of glomerular damage. The system isn't working as it should.
So if the glomerulus is so crucial for filtering, what happens when it's damaged? How do we even know?
That's where something called proteinuria comes in.
Proteinuria. What is that?
It's basically protein in your urine. Remember how the filtration membrane should keep proteins in the blood?
Right, because they're too big to pass through.
Exactly. So if proteins are showing up in your urine, it's a huge red flag.
A sign that something's wrong with the glomerulus.
You got it. It means the filter is broken, letting stuff through that shouldn't be.
And that can indicate serious kidney disease, right?
Absolutely. It's often one of the first signs doctors look for.
Okay, so proteinuria is about the filtration unit. But what about that ischemia we talked about earlier?
Prolonged ischemia, meaning no blood flow for too long, can lead to something even worse for the kidney.
Which is?
Tubular necrosis. Remember those tubules where reabsorption happens?
The tiny tubes that fine-tune the urine?
Yeah, if they don't get blood, their cells die, and that's acute tubular necrosis.
So even if the glomerulus is okay, if the tubules are dead, the kidney's still in big trouble.
Precisely. It can't properly process the filtrate. That's why blood pressure and flow are so critical to monitor.
It all connects, from the tiniest capillaries to the overall function.
It truly does. Every part plays a vital role in keeping us healthy.
So let's kick things off with renal physiology. What's the big picture here?
Okay, so it's all about how your kidneys clean your blood and balance fluids.
Right, like a super advanced filtration system?
Exactly. It starts with glomerular filtration, where blood plasma gets pushed through tiny filters.
And that's where the glomerulus comes in, right?
Yep. The glomerulus is this amazing network of capillaries where the actual filtering happens.
So how does the kidney know what to filter and what to keep?
That's where the juxtaglomerular apparatus is key. It's like the control center, constantly monitoring blood flow.
A control center for filtering?
Totally. It has specialized cells like the macula densa, which senses changes in the fluid passing through.
Macula densa, what does it do?
It's an electrochemical sensor telling the kidney if the filtration rate is too high or too low.
Whoa. so it adjusts things on the fly.
Precisely. And we also have mesangial cells. Short pause. They're like smooth muscle cells.
Muscle cells in the kidney?
Yeah, they can contract or relax to change the surface area for filtration, directly impacting how much gets filtered.
That's wild. What happens after filtration?
Then the fluid moves into the tubular segments, like the proximal tubule, the loop of Henle, and the collecting duct.
And that's where water and other stuff gets reabsorbed, right?
Exactly. An ADH, antidiuretic hormone, plays a huge role in how much water gets reabsorbed, concentrating your urine.
So it's all about balancing filtration, reabsorption, and concentration to keep us healthy.
That's the core of it. It's a remarkably coordinated system.
So after that initial overview, let's zoom in on the glomerulus. What exactly is happening there?
The glomerulus is where the magic of filtration begins, and it all starts with the juxtaglomerular apparatus.
What?
Juxtaglomerular apparatus. It's this cellular complex right next to the glomerulus that produces renin.
Renin, that sounds important.
Super important, and it's linked to something called the macular densa.
The macula densa, another big name.
Think of it as the kidney's own electrochemical sensor. It's in a specialized part of the distal convoluted tubule.
So it's sensing things.
Exactly. It's constantly monitoring the fluid and then regulates the glomerular filtration rate, or GFR, based on what it finds.
Wow, so it's like a tiny quality control system.
Pretty much. And then you have mesangial cells. They're like smooth muscle fibers.
Muscles in the kidney?
Yep. When they contract, they constrict the glomerulus, which reduces the GFR.
And if they dilate?
They expand it, increasing the GFR. It's a precise control system.
That's incredible. So what's actually doing the filtering?
That's the glomerular capillaries. They have 3 layers. First, the endothelium, which is fenestrated, meaning it has little pores for water and solutes.
Okay, so things get through there easily.
Then the glomerular basement membrane. This layer has a negative electrical charge.
Why negative?
It acts as a protein barrier, preventing important proteins from escaping into the urine.
Smart. And the third layer?
That's the epithelium, made of podocytes. They have these amazing foot-like processes that wrap around the capillaries.
Like a net?
Exactly, forming a filtration mesh. They even look a bit like neurons structurally.
No way. Neurons?
Yes, some scientists think they might share a common origin. These podocytes form what we call pre-urine.
Pre-urine?
Before it becomes actual urine. This pre-urine is collected by the Bowman's capsule, a cup-like structure surrounding the glomerulus.
So if any of these parts get damaged,
Then the kidney's ability to filter is severely impaired and homeostasis can go out of whack.
so after all that intricate filtration in the glomerulus, where does this pre-urine go next?
It heads into the tubular system. This is where the real fine-tuning happens.
Fine-tuning how?
Well, we have different segments, each with a specific job for reabsorbing things your body still needs or getting rid of waste.
Okay, so what are these segments?
First, the proximal convoluted tubule. That's where most of the good stuff like sodium, chloride, magnesium, potassium, and water gets reabsorbed.
A lot happens there. And then?
Then it moves to the loop of Henle. This is crucial for concentrating the urine using something called the countercurrent mechanism.
The loop of Henle. Is that different for different glomeruli?
Great question. Yes, there are 2 types. Superficial glomeruli have short loops of Henle.
And the other type?
Juxtamedullary glomeruli. They have these really long loops that dip deep into the kidney.
So longer loop means more concentration power?
Exactly. After the loop, it's the distal convoluted tubule for more ion reabsorption and potassium secretion.
And finally?
The collecting duct. This is the last stop for water reabsorption, heavily influenced by ADH.
ADH, the hormone we mentioned earlier?
Precisely. It dictates how much water gets pulled back, really concentrating that urine.
So each part has its own role in getting the fluid just right.
Absolutely. It's a complex, coordinated effort to maintain balance.
So we've talked about the different parts of the tubules, but how do the kidneys actually concentrate urine?
That's where the countercurrent mechanism in the loop of Henle comes in. It's really clever.
Countercurrent? What does that mean?
Think of it like this. The descending part of the loop is permeable to water.
Okay, so water leaves the filtrate there?
Exactly, making the fluid inside super concentrated.
And then the ascending part?
That part's impermeable to water, but it actively pumps out ions like sodium and chloride.
So it dilutes the fluid again.
Yes, but by pumping out those ions, it creates a super salty environment outside the loop.
Ah, like a gradient in the kidney.
Precisely. This osmotic gradient is critical for the collecting duct later on.
And that's where ADH comes in, right?
You got it. ADH, antidiuretic hormone, acts on the collecting duct.
What does it do?
It basically tells the collecting duct to open up water channels called aquaporins.
Aquaporins, so more water can leave the urine?
Yes, and be reabsorbed back into the body thanks to that gradient the loop of Henle created.
So ADH helps us hold on to water when we need it.
That's why it's antidiuretic. It prevents too much peeing.
It's also coordinated. Glomerular filtration, then all this reabsorption in the tubules.
And ion reabsorption, like sodium and chloride, happens all along the proximal and distal tubules, too.
So the kidneys really are master multitaskers, keeping everything balanced.
Absolutely. It's a symphony of sensors, contractile cells, and hormones, all for homeostasis.
So let's kick things off with the nephron. What exactly is it and what are its main parts?
The nephron is the kidney's fundamental unit. It's like a tiny, intricate filter.
A filter for what?
For blood, it processes blood to form urine, removing waste.
That's wild. So it's basically doing all the work in our kidneys?
Precisely. And it's packed with key structures like the Bowman's capsule.
Bowman's capsule. That sounds important.
It's where filtration begins. Blood enters and gets filtered there.
Okay, so filtration starts, then what?
Next up is the proximal convoluted tubule, PCT for short.
PCT. What does that do?
It reabsorbs essential substances back into the blood, like water and nutrients.
So it takes back the good stuff?
Yep. Think of it as a quality control stage. A lot happens there.
And after the PCT?
We hit the loop of Henle. This is where things get really interesting for urine concentration.
Urine concentration. How so?
It creates a crucial osmotic gradient, helping the body conserve water.
So the nephron isn't just filtering, it's also making sure we don't lose too much water?
Exactly. It's a master of balance. Bowman's for filtering, PCT for reabsorbing, and the loop of Henle for concentrating.
That's a lot of action for one tiny unit.
So let's talk about the Bowman's capsule and the proximal convoluted tubule. What's special about their structure?
Well, both have this amazing brush border structure.
A brush border, like little bristles?
Exactly. In the proximal convoluted tubules, it dramatically increases the surface area.
Ah, for absorbing things better.
Precisely. It's all about efficient exchange. And then it just stops abruptly,
Stops where?
right at the beginning of the loop of Henle.
That's so specific. What else is happening in these parts?
Primary cilia, they're like tiny antennas.
Antennas for what?
They sense the urine, its composition, its direction, how fast it's flowing.
Wait, so these little cilia are like constantly monitoring everything?
Totally. They're critical sensors. And when they don't work right,
That causes problems?
big problems. Many kidney diseases are linked to dysfunctional primary cilia.
So they're not just passive structures, they're actively involved in kidney health.
Absolutely. Think of them as the kidney's quality control department.
That's wild. They're telling the kidney what to do based on the urine. Incredible. So it sounds like the PCT is really just a powerhouse for filtering. But then it's not just one uniform segment, right?
Not at all. It's actually divided into 3 distinct segments: S1, S2, and S3.
Okay, like different phases of a race?
Exactly. Each has a slightly different role.
Tell me more about S1.
S1 is the initial segment. It's packed with mitochondria. Think of it as the muscle of the PCT.
mitochondria means energy, right?
You got it. Lots of energy for active reabsorption of sodium and chloride.
So it's actively pulling important stuff back in.
Precisely. It's super efficient.
And S2?
S2 is similar to S1, still rich in mitochondria, continuing that active reabsorption.
So S1 and S2 are like the heavy lifters.
Yep, they do most of the work.
What about S3 then? Is it also full of mitochondria?
No, actually. S3 has fewer mitochondria.
That's a surprise.
It is. By the time the filtrate reaches S3, most of the sodium and chloride reabsorption has already happened upstream.
So S3 is more like a cleanup crew.
Exactly. It's finishing up the process with less active transport needed.
Okay, so we've covered the Bowman's capsule and the proximal tubule. What happens next?
We're heading into the Loop of Henle, right?
Exactly. This is where things get really cool for urine concentration. Think of it as a U-shaped pipe.
A U-shaped pipe. Got it.
It has a descending thin limb, then the U-turn itself, and an ascending limb that has both thin and thick parts.
So different sections, do they all do the same thing?
No way. Each part is specialized. The descending thin limb is super permeable to water.
So water just leaves there?
Yes, but not much salt. Then you hit the ascending thin limb and it's the opposite. Impermeable to water, but salts move out.
That's a clever design. What about the thick ascending part?
That's the real workhorse for salt transport. It actively reabsorbs sodium, potassium, and chloride.
So it's pumping out all those electrolytes?
You got it. And it's also impermeable to water. All this differential permeability is key
Why is it so key?
because it creates this amazing osmotic gradient in the medulla of the kidney. It's essential for concentrating the urine later on.
So basically the Loop of Henle is building this salty environment around itself?
Precisely. It's setting the stage. Without this gradient, we couldn't make concentrated urine.
That's fascinating. So it's all about making the fluid around it more concentrated so water can be pulled out efficiently?
Exactly that. It's a masterclass in fluid dynamics and reabsorption.
So after all that filtering and concentrating in the loop, where does the fluid go next?
It hits the collecting duct. This is the grand finale for urine concentration.
The grand finale? What makes it so special?
Its structure is fascinating. It's a single layer of epithelial cells. And here's the cool part. It has these things called aquaporins.
Aqua what now?
Aquaporins. They're like water channels. They allow a massive amount of water to be reabsorbed back into the body.
So that's how we avoid dehydrating ourselves every time we pee, right?
Precisely. And these aquaporins are regulated. Guess by what?
Some kind of hormone, I bet.
You got it. Vasopressin. The collecting duct has receptors for it on its basolateral side.
So vasopressin tells the duct, hey, open those aquaporins, we need water.
Exactly. And just like the PCT, the collecting duct also has sensory cilia.
So it's also checking the urine's contents?
Yes, it acts like a little chemical and flow sensor. Super important.
That's wild. How does it compare to the PCT we talked about earlier?
Big differences. PCT has a polarized, multi-layered epithelium. The collecting duct is single-layered.
And the aquaporins?
Limited aquaporins in the PCT, but a ton in the collecting duct. And PCT has no vasopressin receptors.
So they both have cilia but handle water very differently.
Let's jump right in. What exactly are kidneys doing for us?
Well, imagine your body as a super complex, always running factory. Your kidneys are like the ultimate quality control and waste management team.
A factory? I like that. So they're cleaning things up?
Exactly. But it's way more than just cleaning. They're constantly working to keep everything in perfect balance, what we call homeostasis.
Homeostasis. Right. So like the perfect internal environment.
Yes. Think of it like a meticulous chef balancing flavors. They filter blood, regulate fluids, and manage electrolytes.
Electrolytes like sodium and potassium.
Spot on. These are critical for every cell. And they also handle our acid-base balance.
Acid-base balance. That sounds complicated.
It is, but it's crucial. Without it, our body just can't function right. The kidneys are these unsung heroes silently working to keep us alive and well.
So they're filtering, balancing electrolytes, and managing acid. That's a lot for two small organs.
Absolutely. Their roles are so vital, affecting everything from blood pressure to bone health and even red blood cell production.
Wow, so much more than just making pee.
Precisely. They're like the master regulators.
So the kidneys are these master balancers, but how do they actually clean our blood? What's their cleaning superpower?
That's their depurative function. It's basically a 3-step process to get rid of waste like urea.
3 steps? Tell me everything.
First, it's glomerular filtration. Plasma gets filtered, creating this pre-urine.
Pre-urine, like a rough draft.
Exactly. Then tubular reabsorption kicks in. The good stuff, water, nutrients, useful ions, they get reabsorbed back into the body.
So it's not just flushing everything out, it's smart.
Super smart. And the final step is tubular secretion, where the tubules actively push out any remaining waste.
Like drugs or excess ions.
Yep. Anything the body doesn't need, out it goes.
What happens if the system breaks down?
That's when you get chronic kidney failure. All those toxic waste products build up, essentially poisoning the blood.
No way, that's serious.
It is. It highlights just how critical these steps are for our health.
So it's not just about filtering, right? Kidneys also balance the fluids in our body?
Absolutely. They're like the ultimate fluid managers. Think about it. Intravascular volume, that's blood.
Okay, blood in the vessels.
Exactly. And then extravascular, which is interstitial fluid.
Like between the cells?
Spot on. The kidneys are constantly deciding whether to hold on to water and sodium or get rid of them to keep these volumes just right.
And electrolytes too, I'm guessing, like sodium and potassium?
Oh, totally. They're crucial for everything. Sodium, potassium, chloride, calcium, all managed by the kidneys.
Why are those so important?
They affect cell function, nerve signals, muscle contractions, everything. Like sodium is key for extracellular fluid.
So if kidneys mess up, cells get confused?
Pretty much. And potassium is a big deal inside the cells for specific functions. The kidneys adjust what's outside, which indirectly impacts what's inside the cells.
Wild. So they're like the conductors of our body's electrolyte orchestra.
The absolute conductors.
So it sounds like the kidneys are doing a lot of filtering and balancing. What about plasma osmolarity?
Oh, this is where it gets really cool. Plasma osmolality is essentially how concentrated your blood is.
Like how much stuff is dissolved in it?
Exactly. It's the number of dissolved particles per kilogram of solvent, usually water. Not to be confused with osmolarity, which is per liter.
Okay, subtle but important difference. What's the normal value for this?
Around 285 to 295 milliosmoles per kilogram. Keeping it in that range is vital.
So if it changes, what happens?
If it goes up, meaning your blood is too concentrated, your brain triggers thirst.
To make you drink water.
Yep, and if it drops, like your blood is too diluted, you feel less thirsty and your kidneys ramp up urine production.
To get rid of extra water.
Spot on. So the kidneys working with your thirst mechanism constantly adjust water excretion to keep that balance.
So the kidneys are really the mean players here, even though thirst helps.
They are the primary effectors. Intestines and sweating play minor roles, but kidneys really control it through concentrating or diluting urine.
So the kidneys are super precise about osmolarity, right?
Absolutely. They're also really precise about how much water and electrolytes they excrete.
How do they know when to keep water and when to let it go?
Hormones, ADH and aldosterone are key players here.
ADH and aldosterone, they tell the kidneys what to do?
Exactly. They signal the tubules to either reabsorb more water or excrete it.
And that helps control our overall fluid volume.
Yes, and it's also linked to plasma volume.
So water and electrolytes like sodium and potassium are tied together?
Totally. Electrolytes like sodium, potassium, chloride, calcium, they're not just important nutrients.
They're osmolytes, right?
You got it. They directly impact plasma osmolality.
So the kidneys manage their excretion and that controls how concentrated our blood is?
Precisely. It's
all about balance to keep your system running smoothly.
Okay, so kidneys do all this amazing stuff with water and electrolytes, but what about acid-base balance? That sounds super important.
Oh, wait, absolutely is. Think of it like a delicate tightrope walk with hydrogen cations, that's H+, and bicarbonate anions.
H plus and bicarbonate, got it. So the kidneys are walking at tightrope?
Exactly. They work hand in hand with your lungs to keep everything perfectly neutral.
The lungs too? How do they fit in?
Well, your lungs get rid of CO2. That's a major player in this whole acid-base system.
Right, breathing out carbon dioxide.
And the kidneys, they can either get rid of excess acids or ammonia, depending on what the body needs.
They adjust their acid-removing superpowers on demand?
Pretty much, they control those H+ and bicarbonate levels precisely.
Wait, are there other things that help too?
Yep, your bones actually store phosphate ions that can buffer, and even albumin, a protein, can snatch up H+.
No way, bones too? So if your kidneys fail?
Then things go really wrong. They can't get rid of enough acid.
Which means...
You end up with metabolic acidosis. It's not good.
So everyone knows kidneys filter waste, right? Like a coffee filter for your blood.
Exactly. But they do so much more than just filter. They're actually endocrine powerhouses.
Endocrine? Like hormones?
Precisely. They produce or activate crucial hormones that impact your entire body.
Wait, really? What kind of hormones are we talking about?
Well, there's erythropoietin, vitamin D, and a whole system called RAAS.
RAAS? Sounds complicated.
It's basically how they regulate blood pressure and fluid balance. Super important.
So it's not just about peeing out toxins then.
Not at all. They're like little mini factories, constantly signaling and coordinating.
That's wild. So if your kidneys aren't working right, it's not just waste buildup.
Exactly. Hormone imbalances can cause a cascade of problems, far beyond just filtration issues.
So these hormones, they're like the unsung heroes of kidney function.
Totally. They keep everything in balance even when you don't realize it.
So let's talk about EPO. Erythropoietin, what even is that?
It's a hormone your kidneys make. It tells your bone marrow to pump out more red blood cells.
Like a signal?
Exactly. More red blood cells means more oxygen delivered everywhere.
So if your kidneys aren't working right?
Less EPO, and that means anemia. It's a common problem with kidney failure.
Oh, so people with chronic kidney disease, they get anemic because of this.
Yes, it's a persistent anemia, and it's a big deal.
So how do they fix it?
They can get synthetic EPO. It's a game changer for those patients.
Wait, so we can just make it?
We can. It replaces what their kidneys can't produce anymore.
That's incredible. So it's not just about filtering waste, it's about making vital hormones too.
Absolutely. Kidneys are endocrine powerhouses.
So beyond red blood cells, kidneys are also like super important for our bones, right? With vitamin D.
Absolutely. It's one of their biggest endocrine roles. The kidney actually activates vitamin D3.
Activates it? So it's not active when we get it.
Not fully. Your liver first processes it into 2,5-hydroxycholecalciferol.
How mouthful.
It is, but then the kidney takes that and turns it into the super active form, 1,25-dihydroxycholecalciferol.
And that's the one our bodies really use?
Yes, this active vitamin D is crucial for absorbing calcium from your gut.
Okay, so if your kidneys aren't working right...
Exactly. Less active vitamin D means less calcium absorption. Then your body starts pulling calcium from your bone.
No way. So it weakens them.
Totally. It can lead to osteodystrophy, which is basically bone damage because of kidney failure.
That's wild. So they give you synthetic vitamin D for that?
They do. Active vitamin D supplements are a common treatment.
So the kidney's like a bone protector too.
In a way, yes. It keeps your calcium levels balanced for strong bones.
So we've talked about hormones and bone health. What about blood pressure regulation? How do kidneys play a role there?
That's where the renin-angiotensin-aldosterone system, or RAAS, comes in.
RAAS. That sounds like a super important system.
It really is. It's a whole cascade that controls your blood pressure and fluid balance. Think of it like a finely tuned thermostat for your body's fluid.
A thermostat for blood pressure. So how does it start?
The kidney releases this enzyme called renin.
Renin. Okay.
Right, and renin chops up a protein from your liver called angiotensinogen into angiotensin 1.
Chopping it up.
Then in your lungs, another enzyme converts angiotensin 1 into angiotensin 2.
So it's a two-step conversion process.
Exactly. And angiotensin 2 is the start here. It's a powerful vasoconstrictor, meaning it narrows blood vessels.
So narrow vessels means higher pressure.
Spot on. And it also tells your adrenal glands to release aldosterone.
Aldosterone, what does that do?
Aldosterone makes your kidneys reabsorb more sodium, and with sodium, water follows.
More water means more blood volume and even higher pressure.
You got it. It's a brilliant but complex system to keep things in check.
Okay, so we've talked about all these hormones, but how does the kidney actually physically clean the blood?
That's where the glomerulus comes in. It's the kidney's primary filtering unit.
The glomerulus, what even is that?
Think of it as this tiny, super intricate ball of capillaries. It's inside something called Bowman's capsule.
A ball of capillaries? Like a little super dense spaghetti ball.
Exactly, and its job is huge. It takes the plasma from your blood and removes a bunch of water.
So it's filtering out water and stuff.
Yep. This process is called ultrafiltration, and what's left is this ultrafiltrate rich in electrolytes, toxins, all that good stuff.
So it's basically the first step to cleaning everything up.
Spot on. Without it, the other processes we talked about wouldn't even have anything to work with.
That's wild. It's like a tiny, super efficient coffee filter for your blood.
Precisely. It's constantly working to maintain balance.
So how efficient are these tiny filters then? We're talking glomerular filtration rate, GFR.
Super efficient. It measures how much plasma the glomerulus filters per minute.
And what's a typical number?
Around 120 milliliters per minute. That's about 170 liters of filtrate a day.
Wait, 170 liters? But we only pee out 1 or 2 liters daily.
Exactly. That's the crazy part. Over 99.9% of that filtrate gets reabsorbed.
So the tubules are basically super thirsty sponges.
You got it. They pull back almost everything your body needs.
That's incredible. So GFR is a huge indicator of kidney health, right?
Absolutely. It's the gold standard for measuring kidney function.
Because if that filtration rate drops, bad stuff starts building up.
Precisely. And that massive filtering isn't even for the kidney's own needs.
It's all for the rest of us.
Yep, to keep your whole body's internal balance.
So let's jump right into glomerular pressure. What exactly is it?
Think of it as the net force that pushes fluid out of your kidney capillaries. It's all about filtering your blood.
Okay, so like a tiny coffee filter for your body?
Precisely. And ultrafiltration is that actual process, the liquid moving through the filter.
Got it. So what are these forces? Are there good guys and bad guys?
Kind of. There are forces that favor filtration and forces that oppose it.
Like a push and pull?
Exactly. The main one pushing is the glomerular capillary blood pressure. It's the pressure from the heart driving blood into those tiny filters.
So higher blood pressure means more filtration.
In a simplified way, yes. Then there's also the Bowman's capsule colloid osmotic pressure, but that's usually almost zero because there are no proteins there.
Why no proteins?
Because a healthy filter keeps them out. The pre-urine should be protein-free.
Makes sense. And the opposing forces?
You have the hydrostatic pressure in Bowman's capsule. That's the fluid already filtered, pushing back. And also the colloid osmotic pressure in the glomerular capillaries.
That's the proteins trying to hold on to the water, right?
You're spot on. It's due to the plasma proteins, mostly albumin, in the blood.
So how exactly does this ultrafiltration pressure get, determined? What are the big players?
Right. Think of it like a tug of war. We have forces pulling liquid out and forces pushing it back in.
Tug of war. I like that. So what's pulling it out?
The main one is glomerular capillary blood pressure. It's the brute force, literally pushing the fluid out of the capillaries.
Okay, so blood pressure is a big deal here. What else helps?
There's also Bowman's capsule colloid osmotic pressure, but that's usually almost zero.
0. Why is that?
Because the pre-urine in Bowman's capsule shouldn't have proteins. No proteins, no osmotic pool from them.
Makes sense. So those are the good guys favoring filtration. What are the bad guys trying to stop it?
First, we have hydrostatic pressure in Bowman's capsule. It's the pressure of the fluid already in the capsule trying to resist more coming in.
Like a full balloon resisting more air?
Exactly. And then the big one opposing filtration, colloid osmotic pressure in the glomerular capillaries.
Is that because of the proteins?
Yes, those plasma proteins, mainly albumin, stay in the blood and pull water back towards them, opposing the filtration.
So it's all about who's pulling harder, essentially.
Precisely. The net result of these pushes and pulls determines our ultrafiltration pressure.
So if these forces are constantly at play, how does this whole process evolve as blood moves through the glomerular capillary?
That's the fascinating part. Imagine the blood entering the capillary.
Okay, I'm picturing it. What's it like at the start?
At the very beginning, your blood is about 40 to 45% cells, the hematocrit, and 55 to 60% plasma.
And that plasma has all the proteins, right?
Exactly. Now, as water gets filtered out into Bowman's capsule, what happens to the proteins left behind in the capillary?
Oh, they get more concentrated, like boiling down a sauce.
You got it. So the colloid osmotic pressure inside the glomerular capillaries, that pi GC we talked about, starts to skyrocket.
Wait, so it gets stronger and stronger as it goes along?
Yes, absolutely. It increases so much that eventually it can actually equal the net outward pressure.
And when that happens?
Filtration stops. It literally halts.
So the filtering isn't happening all the way through the capillary.
Not continuously. It's highest at the beginning and then tapers off until it stops.
That's wild. So not all glomeruli are always on then.
Precisely. Some can even be at rest and get recruited when needed, like if you lose a kidney.
Like a backup team waiting to jump in.
Perfect analogy. It's all about efficient regulation.
So after all these forces we've talked about, how do we actually measure the filtration rate?
That's where the single nephron GFR, or SNGFR, comes in. It's how fast one nephron filters.
SNGFR. Okay, so what's the formula?
It's SNGFR equals KF times PUF.
KF. What's KF?
That's the ultrafiltration coefficient. It's super important
Why is it so important?
because it tells us about the hydraulic permeability of the capillaries and the actual filtration area. Think of it as how leaky and big the filter is.
So KF is like the efficiency of the filter.
Precisely. A high KF means more fluid to pass through. It's a combination of permeability, LP, and the surface area, A.
Got it. So we have the filtration rate for one nephron, but how do we get the total GFR for the whole kidney?
You just add them all up. The total GFR is the sum of all the SNGFRs from every single nephron.
Wow. How much is that normally?
About 170 liters per day. That's a lot of fluid being filtered.
170 liters?
That's insane, right? Your kidneys are constantly working overtime.
So the KF and the PUF are key to that massive filtration.
Absolutely. Any changes in those and your GFR changes dramatically.
So that brings us to the glomerular basal membrane. What exactly is its job in all this?
It's like a super smart filter, right? It's a hydrophilic gel, but it's also electrostatically charged.
Electrostatically charged? Like a magnet?
Pretty much. It actively repels negatively charged plasma proteins, like albumin, but lets water through easily.
So it's specifically designed to keep the good stuff in your blood, but let the waste out.
Exactly. It's super selective.
And how does aging affect this amazing filter?
Well, as we get older, this membrane can thicken and become fibrous.
So it gets stiffer, like an old sponge.
Yep. That thickening actually decreases its hydraulic permeability, the KF we talked about.
Which means it doesn't let as much through.
Precisely. The filtration flow goes down, and so does your GFR.
So basically, the older you get, the less efficient your kidneys become at filtering.
In a nutshell, yes.
A young, thin membrane means high permeability and normal GFR, around 170 liters a day.
But an older, thicker membrane means lower permeability and a reduced GFR.
That's why kidney function can naturally decline with age.
Fascinating how something so microscopic has such a big impact on our overall health. So let's kick things off with the absolute basics. What exactly is renal filtration?
Okay, so renal filtration is basically the kidney's way of separating your blood from what will eventually become urine.
Wait, like a coffee filter for your body?
Exactly. It's a super critical process that keeps us healthy, getting rid of waste, but keeping the good stuff.
And where does this magic happen?
It all starts in the glomerulus. Think of it as this tiny, super efficient processing plant inside your kidneys.
So the glomerulus is where the filtering begins.
Yes, and it's surrounded by something called Bowman's capsule. Together, they form this amazing filtration barrier.
A barrier that decides what stays and what goes?
Precisely. It's an ultra microscopic structure that's super selective about what gets through from your blood into the pre-urine.
Pre-urine. That's a cool term.
It is. It's the fluid after initial filtration, but before it's been fully processed by the tubules.
So this barrier has different parts then?
Absolutely. It's not just one thing. It's a 3-layered masterpiece that we'll dive into next.
3 layers? That sounds complex.
It is, but each part plays a specific role in keeping those vital proteins in your blood while letting waste products pass.
So after that initial filtration, what actually makes up this filtration barrier itself? I mean, how does it physically do its job?
Right, it's super intricate. Think of it like a 3-layer security system for your blood. First up, you have the fenestrated endothelium.
Fenestrated, like windows.
Exactly. It's this capillary network with tiny pores. It lets a lot of fluid and small solutes through, but keeps the blood cells in.
Okay, so like a first pass filter. What's next?
Then you hit the glomerular basement membrane. This is a special hydrophilic gel layer that wraps around the capillary wall.
A gel. What does that do?
It's like a sticky net. It's negatively charged, which is key for repelling negatively charged proteins, keeping them out of the pre-urine.
Oh, so it's not just size, it's also about electrical charge.
Totally. And then for the final layer, you have the podocyte foot processes.
Podocytes. That sounds like something from a sci-fi movie.
They're these super cool epithelial cells with these finger-like projections. They interdigitate, forming these tiny slit pores.
So it's like a final super fine mesh.
Precisely. They form the most selective part of the barrier. It's how our bodies ensure only the right stuff passes into the pre-urine.
That's incredible. All those microscopic parts working together to keep things just right.
So after all that filtering, how does the kidney decide what proteins actually get through?
That's the fascinating part. It's super selective, almost like a bouncer at a club.
A protein bouncer? Tell me more.
Exactly. It filters based on 3 main things: charge, molecular mass, and diameter.
So like big, bulky, positively charged proteins are out?
Well, actually, it's the opposite for charge. Only positively charged molecules can pass easily.
Wait, really? I would have thought negative charges would sneak through.
No, because the filtration barrier itself has a negative charge. It repels other negative proteins, which is clever.
Ah, so negative repels negative. Makes sense.
Yep. Then there's molecular mass. Think of it like a weight limit.
So heavier proteins get stopped?
Pretty much. And diameter is the final gate. If you're too wide, you're not getting through those tiny pores.
It's like a strict dress code and a size limit for proteins.
Perfect analogy. This multi-layer selection ensures only the right stuff gets into the pre-urine.
That's incredibly precise. It's not just deceive, it's a smart filter.
Exactly. It's why we don't usually find large proteins in our urine. The kidney is super efficient at keeping them in the blood.
So after all that intense filtering, what happens to the stuff the body actually needs?
That's where tubular reabsorption comes in. It's like the kidney's recycling plant.
Recycling? So it takes back the good stuff.
Exactly. It reclaims essential molecules and electrolytes from the pre-urine, sending them back to the blood.
Like what?
Things like sodium, chloride, potassium, but also calcium, magnesium, even amino acids, and all the glucose.
All the glucose? Wow.
Yes, pretty much all of it. And it mostly happens in the proximal tubule, which has these amazing brush borders.
Brush borders. Like a tiny broom?
Kind of. They're microvilli that drastically increase the surface area for absorption. Super efficient.
So how does it get everything back?
Through both active and passive transport. Active transport, like the sodium-potassium ATPase pump, uses energy.
It needs energy for this?
A lot of it. It's constantly pumping sodium out of the tubule into the blood. That's why kidneys are so sensitive to oxygen deprivation.
Oh, right, because no oxygen means no energy.
Precisely. And passive transport, like chloride and potassium, just follow electrical gradients.
So sodium goes out and then chloride follows it.
Yes, because sodium is positive and attracts the negative chloride. Together they form salt, which pulls water along with it.
To maintain neutrality?
Exactly. And potassium follows suit. It's all about balancing everything out.
Okay, so after all that reabsorption, what about specialized channels and transporters that are involved?
Oh, these are crucial. We're talking about things like aquaporins and solucarrier transporters.
Aquatic porins? That sounds like water pores, right?
Exactly. They're literally water channels, super important for letting water move freely in and out of cells.
So just for water?
Mainly, yes, but then you have these other big players called SLC transporters, solute carrier transporters.
SLC, what do they do?
They're a huge family of membrane proteins doing everything from moving nutrients to maintaining pH.
pH, like that Na plus H plus exchanger we just mentioned?
Spot on. That's an SLC transporter, crucial for acid-base balance in the body.
Wow, so these specialized channels are literally keeping us alive?
Absolutely. They're like the finely tuned mechanisms that ensure everything stays in perfect physiological homeostasis.
And are there others?
Oh yeah, like the chloride bicarbonate exchanger, swapping two anions without needing energy, helping with pH too.
So many moving parts just to make pee.
It's way more complex than just making pee. It's about filtering and rebalancing every second.
Mind blown. This is incredible. Well, that's a lot to think about.
Here.
Okay, so we're talking kidneys, right? And sodium ions are super important there.
Absolutely. Sodium movement is key to almost everything the kidneys do, especially with fluid balance.
So how do these sodium ions actually move around inside the kidney cells?
Well, the star of the show is the sodium-potassium ATPase pump.
The ATPase pump, like a little engine?
Exactly. It uses ATP to swap 3 sodium ions from inside the cell for 2 potassium ions outside.
So it's constantly pumping things?
Yes, creating this electrochemical gradient that powers other transport mechanisms.
Ah, like a domino effect for sodium.
Precisely. Then we have antiports, like the sodium-hydrogen antiport.
Antiport? So one goes in, one goes out?
You got it. It swaps 1 internal sodium for 1 external hydrogen ion, helping reabsorb sodium and acidify urine.
Acidify urine? Wait, that's important too?
Crucial for maintaining the body's pH balance. And there's also the sodium-calcium antiport.
That one moves calcium?
It swaps 3 sodiums for 1 calcium, creating an electrogenic current because the charges aren't balanced.
So electricity is involved there?
Indeed. Then we have the ENaC channel for epithelial sodium channels.
ENaC? Is that like a gate for sodium?
A gate, but it's regulated by aldosterone, super important for fine-tuning sodium reabsorption in the distal tubule.
Aldosterone controls it, like a hormone.
Exactly. And we can't forget cotransporters, like the sodium-potassium 2-chloride cotransport.
That's a mouthful. What's it do?
It moves sodium, potassium, and 2 chlorides all together, keeping everything charge neutral. Super important in the loop of Henle.
Wow, 4 ions at once?
Yep, and there's also the simpler sodium chloride cotransport in the distal tubule.
Just sodium and chloride.
That's right, reabsorbing them together. All these mechanisms work in concert.
So these sodium transporters are super important, but where do all these amazing things happen in the kidney?
That's where the segmented structure of the renal tubule comes in. Think of it like a super specialized factory line.
A factory line for filtering our blood? That's wild.
Exactly. Each part of this line, the nephron, has a very specific job to do.
Okay, so what's the first stop on this kidney factory tour?
First, you have the convoluted tubule, which is supercoiled, and then the loop of Henle.
The Loop of Henle? That sounds like a roller coaster for fluids.
It kind of is. It has three main parts: the descending limb, the true loop itself, and the thick ascending limb.
So different parts, different jobs.
Totally different. Like the descending limb is all about letting water out, making the fluid more concentrated.
And the ascending limb?
It's the opposite. It's practically waterproof, but actively pumps out salts.
Whoa, so it's setting up this amazing balance.
Precisely. After the loop, you hit the distal convoluted tubule.
Another convoluted tubule?
Yes, but this one is more for fine-tuning. And finally, the collecting ducts. These are the last stop.
So from initial filtering all the way to, well, you know.
Yep, to ushering the final urine out. Each segment specialization is critical for creating the osmotic gradient.
The gradient for what?
For concentrating our urine, which is what we'll talk about next with the countercurrent mechanism.
Okay, so we've got these specialized parts of the nephron, right? That brings us to the countercurrent concentration mechanism.
Yes, this is where it all comes together to concentrate your urine. It's really clever.
So it's all about concentrating urine.
Exactly. The goal is to concentrate solutes like urea in the renal medulla and dilute them in the cortex.
Like making it super salty in one place and less salty in another?
That's a good way to put it. It starts with the fluid entering the loop of Henle, which is isotonic with blood.
Meaning it has the same saltiness.
Right. But then it hits the descending limb of the loop.
The part that's super permeable to water but not solutes, correct?
Spot on. So water rushes out into that salty medulla we just talked about because of osmosis.
Making the fluid inside the tubule even saltier as it goes down?
Precisely. Its osmolarity can shoot up from 300 to 1200 milliosmoles as it descends.
That's a huge change.
Then comes the thick ascending limb. This part is the opposite.
Impermeable to water.
Yes, but it actively pumps out sodium, potassium, and chloride ions.
So it's taking out the salt without taking out the water.
That's it. This dilutes the fluid inside the tubule while making the surrounding medulla even saltier.
And that creates this concentration gradient from the outer cortex to the inner medulla.
Exactly. That gradient is what allows your kidneys to produce urine that's either super concentrated or very dilute, depending on what your body needs.
So that differential permeability and active transport working against each other really drives this whole system. So it sounds incredibly intricate. How do people even begin to visualize something this complex, like the whole countercurrent mechanism?
That's where diagrams become our best friend. They're not just pretty pictures.
They really help break it down?
Absolutely. Think of it like a plumbing system, a network of tubes and valves.
Like a miniature city's waterworks?
Exactly. Each tube represents a part of the nephron, and the arrows show the fluid flowing.
So you can literally see the urine's journey.
Yes, from the initial filtration all the way to the collecting ducts.
And the changes in osmolarity we talked about earlier?
Diagrams often use color gradients or numbers to show how concentrated the fluid becomes.
That makes so much sense, like a heat map for saltiness.
Precisely. And the most powerful part is seeing the countercurrent flow.
Where the descending and ascending limbs are right next to each other?
With fluid moving in opposite directions, allowing that crucial exchange.
Without that visual, it would be so hard to grasp how they influence each other.
It's key to understanding how the kidney achieves such incredible concentration.
So diagrams aren't just for textbooks. They're essential tools for learning this stuff.
Couldn't agree more. They simplify the complex, making the invisible processes of our body visible.
That's pretty cool, like having X-ray vision for your kidney. Let's zoom in on the Henle loop. What exactly is its main gig in the kidney?
Its main gig? It's like the kidney's personal water filter, but with a twist. It concentrates and dilutes the filtrate.
Concentrates and dilutes? At the same time?
Not exactly at the same time, but it has different segments for each job. Think of it like a two-part process.
Okay, 2 parts. So how does it start this concentration game?
The first part is the descending segment. This part is super permeable to water.
So water just rushes out?
Precisely. It's like a sponge releasing water, but it's passive. The water leaves, but the solutes like sodium, potassium, and urea, they stay put.
Wait, so they get stuck inside?
Exactly. They stay in the tubule, making the fluid inside super concentrated.
That's wild. So the fluid gets saltier and saltier.
Yes, much saltier. It can reach an osmolarity of like 1,200 milliosmoles per liter in a healthy person.
Wow. That's a lot. And what about the other segment?
That's the ascending segment. It's completely different.
How so?
This part is impermeable to water. Water cannot escape here.
No water out. So what happens instead?
It's all about solute removal now. It has these special pumps and co-transporters, like the sodium potassium 2-chloride co-transporter.
So it's actively kicking out the salt?
You got it. It actively pumps out sodium, potassium, and chloride into the surrounding tissue.
Making the fluid inside less salty now.
Spot on. By the end of the ascending limb, the fluid's osmolarity drops to around 100 milliosmoles per liter.
From super salty to barely salty. That's a huge shift.
It is. It sets up this incredible osmotic gradient, which is crucial for everything else the kidney does.
So the descending limb concentrates and the ascending limb dilutes, creating this gradient?
Exactly. It's like a finely tuned machine preparing for the next steps in water balance.
So that Henle loop is setting the stage. How does this countercurrent mechanism actually work to create that osmotic gradient?
Is super cool. Think of it like a clever little exchange system.
An exchange system?
Yeah, you've got the descending limb and the ascending limb of the loop running parallel but in opposite directions.
Okay, so like two pipes next to each other, water flowing opposite ways.
Exactly. In the descending limb, water is reabsorbed.
Pulled out?
Yep. The tube is permeable to water there. So as the filtrate goes deeper, more water leaves it.
Making it more concentrated.
Precisely. But here's the trick. The ascending limb is impermeable to water.
Wait, so water can't get out there?
Right. Instead, it actively removes solutes like sodium, potassium, and chloride.
Oh, so it's dumping salt and stuff out.
Into the interstitium, the space around the tubules. This creates a really high concentration of solutes there.
Which then pulls more water out of the descending limb.
Exactly. It's a self-reinforcing cycle. The deeper you go into the kidney's medulla, the saltier it gets.
So it's building this osmotic gradient, this difference in saltiness.
Spot on. That gradient is crucial for pulling out as much water as possible later.
So basically, the descending limb concentrates by losing water, and the ascending limb dilutes by losing solutes, and that solute loss makes the outside super salty.
You got it. That concentrated medulla is the key. Without it, we couldn't make concentrated urine.
And that's why long loops of Henle are so important for animals in dry climates, right?
Absolutely. Longer loops mean more surface area, more time for that exchange, leading to a steeper, more effective gradient.
That's wild. It's like a natural engineering marvel inside us. So we've built this amazing gradient in the kidney, right? How does that actually help us reabsorb water then?
That gradient is everything. Water reabsorption is purely passive, meaning it just follows where the salt is more concentrated.
So it's like a magnet pulling the water out?
Exactly. It's pulled out of the tubules and back into the body, driven by that osmotic gradient created by the Henle loop.
And how does the water actually get through the cell membranes?
That's where aquaporins come in. They're these special protein channels specifically for water.
Like little water tunnels?
Perfect description. Highly selective tunnels for water to pass through quickly.
Okay, but what if you're like super dehydrated? Does your body just hope there are enough aquaporins?
No way. That's where ADH, anti-diuretic hormone, becomes the superstar.
ADH? I've heard of that. The anti-pee hormone?
Pretty much. It's produced by your hypothalamus and released when your blood gets too concentrated.
So when you need to save water?
Precisely. ADH tells the collecting tubules to insert more aquaporins into their membranes.
More tunnels.
More tunnels, meaning more water gets reabsorbed, preventing dehydration. It's a critical mechanism.
So without ADH, you just keep peeing out all your water?
You'd essentially lose too much water, leading to severe dehydration. It's what happens in a condition called diabetes insipidus.
Wow, so this hormone is literally life-saving.
Absolutely. It's the body's ultimate water manager, ensuring we maintain proper hydration levels, especially in challenging environments.
All powered by that initial gradient from the Henle loop. It all connects.
Every single piece works together in this incredible system. Your kidneys are always working for you.
So vasopressin is super important, right? But how does our body actually make it?
It's a fascinating journey, actually. It starts as this huge precursor molecule,
Huge, like how big?
about 164 amino acids long. We call it pre-pro-vasopressin.
Pre-pro-vasopressin. That's a mouthful.
It really is. But it's just a starting point. Think of it like a raw block of marble.
Okay, a block of marble that needs sculpting.
Exactly. The first step is removing a signal sequence. That turns it into pro-vasopressin.
So like cutting off the excess clay.
Precisely. Then there's a crucial enzymatic cut that separates out something called neurophysin.
Neurophysin, what's that for?
It's actually a reserve peptide. It helps store the vasopressin until it's needed.
Oh, so it's not the active hormone itself?
Not yet. After that, there are more modifications, more enzymatic trimming.
More sculpting.
Exactly, until we finally get to the active vasopressin, which is a tiny cyclic peptide.
Tiny? How tiny?
Just 9 amino acids long. So from 164 down to 9.
Wow, that's a huge reduction. So all that processing just to get this small, powerful hormone.
Absolutely. It ensures ultra-regulated control over its availability. Every step is precise.
That's wild. So it's not just made, it's meticulously crafted.
A perfect example of the body's incredible biochemical machinery at work.
So it starts as this giant molecule and gets chopped down to size. Where does this tiny active vasopressin actually do its magic?
Great question. It goes to work in the kidneys, specifically at the vasorecta, which are these deep vessels.
The vasorecta, is that like its target zone?
Exactly. It connects with the collecting ducts there, and it's all about how it binds to this specific receptor.
A receptor, like a lock and key.
You got it. It's called the V1 receptor. When vasopressin, the key, fits into the V1 lock,
Something big happens.
oh, something huge. It kicks off a signaling cascade.
A cascade?
Think of it like a domino effect inside the cell. First, it activates an enzyme called adenylate cyclase.
Adenylate cyclase. What does that do?
That enzyme then massively increases levels of something called CAMP.
CAMP. Why is that important?
Because CAMP is a critical indicator of your hydration status. When you're thirsty, its levels can jump 20 to 30 times.
20 to 30 times. That's wild. So a big jump in CAMP means I'm dehydrated?
Precisely. It's the body's internal alarm bell, signaling, hey, we need more water.
Wow. So vasopressin binding to V1, activating adenylate cyclase, then CAMP surges.
All setting the stage for what happens next to actually conserve that water.
It's all connected.
So we've got all this CAMP, this marker of hydration. What happens next in the kidney?
Well, CAMP acts like a crucial second messenger, activating something called protein kinase A, or PKA.
PKA, what's its job?
It phosphorylates these tiny recycling vesicles that contain aquaporin 2, or AQP2.
Aquaporin 2 sounds important for water.
It absolutely is. Once those vesicles are phosphorylated, they move to the surface of the collecting duct cells.
So they just pop out?
Exactly. They exocytose their AQP2 channels onto the luminal membrane. Think of it like opening up a bunch of tiny water gates.
Water gates. Okay, so now the kidney can reabsorb more water.
Yes, it dramatically increases the water permeability of the collecting duct.
And
that's how we stay hydrated, right, through this whole process.
Spot on. The renal interstitium is naturally hyperosmolar, so water rushes in by osmosis and then goes back into the blood.
That's incredible. How much difference does this actually make?
If this system didn't exist, we'd be peeing out something like 170 liters of urine every single day.
Wait, 170 liters? No way.
It's wild. It's why terrestrial animals evolved this sophisticated system to conserve water.
So our ability to live on land is thanks to these tiny aquaporin channels.
Essentially, without them, we'd quickly dehydrate.
That's a huge evolutionary one then.
A massive one. It's truly amazing what our bodies do to maintain balance.
So we've talked about how vasopressin works its magic, but what happens when something goes wrong with its production?
That's where essential diabetes insipidus comes in, a really serious condition.
Oh, like diabetes but not sugar diabetes?
Exactly. It's completely different. This is all about water balance or the lack of it.
So what causes it?
It usually comes down to mutations in either the vasopressin precursor molecule we talked about or in neurophysin, that storage peptide.
Wait, so if those are messed up, the whole production line breaks down?
Pretty much. Either vasopressin isn't made at all, or it's malformed and can't do its job properly.
And that means no ADH?
Insufficient ADH production, or none at all, so the kidney collecting ducts just can't reabsorb water effectively.
Oh no, so then you're just constantly losing water?
Massive amounts. Patients can urinate huge volumes of very dilute urine, sometimes up to 20 liters a day.
20 liters? That's wild. They must be constantly thirsty.
Uncontrollably so, trying to keep up with the water loss. It's a constant battle against dehydration.
So it's not about blood sugar, but about the body's inability to hold on to water.
Spot on. Short pause. It's a critical malfunction in the body's water regulation system, all because of a tiny mutation in that initial vasopressin factory.
That just shows how vital those early steps in the peptide's journey are.
So let's jump right into these vasopressin receptors. What exactly are they and where do they hang out?
Okay, so vasopressin receptors are super important. They're mainly found in your kidneys, specifically in the collecting ducts, deep inside the nephron.
In the collecting ducts, got it. And what do they do there?
Their job is to respond to a hormone called vasopressin, or ADH. This hormone tells the kidneys to reabsorb water.
So they're like the kidney's water managers?
Exactly. When vasopressin binds to them, it triggers a cascade to put water channels on the surface so the kidney can pull water back into the body.
That makes sense. But what happens when things go wrong, like with mutations?
Ah, that's where it gets interesting. Hundreds of mutations have been identified in these receptors, especially the V2R receptor.
Hundreds? Wow. And what do these mutations lead to?
They prevent vasopressin from binding properly. So no binding, no water reabsorption. It leads to something called nephrogenic diabetes insipidus.
Nephrogenic diabetes insipidus. That sounds serious.
It is. Because the kidney can't reabsorb water, the body loses massive amounts of fluid, causing extreme thirst and frequent urination.
So it's like the kidney just stops listening to the water-saving signals.
Precisely. And it's not just the V2R receptor. Sometimes it's a mutation in something called aquaporin 2 or AQP2.
AQP2. What's that?
That's the actual water channel. If that's mutated, even if the receptor gets the signal, the channels don't get to the cell surface to move water.
So either the signal receiver is broken or the actual water gate is broken.
You got it. Both lead to the same problem. The collecting duct becomes impermeable to water. It's like having a leaky bucket.
That's a great analogy. So two different ways for the system to fail, but both result in the body losing too much water.
Exactly. The V2R receptor mutations are more common, but AQP2 mutations, though rare, also cause this condition.
So these mutations mess up the whole system, but how does water reabsorption even work in the first place? Walk me through the nephron's journey.
Okay, think of it like an assembly line for water. It all starts in the glomerulus.
The glomerulus, that's where the initial filtering happens, right?
Spot on. We filter about 120 milliliters per minute there. The osmolality, that's the concentration, is roughly 300 milliosm per kilogram at the start.
So it's just filtering everything out initially?
Exactly. Then it moves into the proximal tubule. This is where a lot of the work happens.
What kind of work?
Here, about 2/3 of the water gets reabsorbed, along with electrolytes. But the osmolality stays pretty much the same. It's called isosmotic reabsorption.
2/3? Wow, so the concentration doesn't change much, even though so much water leaves.
Nope, because solutes are also being reabsorbed. Next up, the loop of Henle.
Oh, the loop. That's a classic.
It is. The descending limb slightly absorbs water, making the fluid more concentrated, up to about 1,200 milliosam per kilogram.
So it gets saltier there.
Precisely. But the ascending limb, that part is impermeable to water. It only reabsorbs solutes, so the fluid becomes less concentrated.
Ah, making the urine dilute. And then the final stretch?
The distal convoluted tubule and the collecting duct. This is where ADH, antidiuretic hormone, really shines.
The ADH we just talked about, that's tied to vasopressin, right?
The very same. With ADH, rapid water reabsorption happens and you produce concentrated urine, only about 0.3 liters a day.
But without ADH, or if the receptors are broken...
No water reabsorption. You'd be eliminating 20 liters of urine daily. You'd need to drink 20 liters just to keep up.
20 liters? That's insane. So ADH is critical for keeping us from becoming severely dehydrated instantly.
Absolutely. It's the ultimate water-saving hormone.
So after all that, what are the real-world implications of these vasopressin receptors and mutations? Like, what happens when it goes wrong?
That's where diabetes insipidus comes in.
Oh, like diabetes, but it's not about sugar, right?
Exactly. That's the common misconception. This is about water balance. There are two main types, central and nephrogenic.
Okay, so central diabetes insipidus, what's the problem there?
With central, your brain just isn't making enough ADH. No ADH, no signal for the kidneys to reabsorb water.
So like your body can't even tell the kidneys to do their job?
Precisely. The kidneys themselves are fine, but they're not getting the instructions.
And how do you fix that?
We can treat it with something called desamino ADH or DDAVP. It's essentially a synthetic ADH.
So you just give them the hormone their body isn't producing.
Right. It's like replacing the missing messenger.
Makes sense. What about nephrogenic diabetes insipidus?
That's when the ADH is there, but the kidneys aren't responding to it.
Oh, so like the vasopressin receptor mutations we talked about.
Yes, exactly. Either the V2R receptor's faulty or the aquaporin 2 channels aren't working right.
So the instructions are sent, but the kidneys can't hear them or can't follow them.
Exactly. The collecting ducts stay impermeable to water.
And how do you treat that? You can't just give them more ADH if the kidneys aren't listening.
That's the challenge. The treatment is different. We use thiazide diuretics, a low-sodium diet, and careful hydration management.
Diuretics? Wait, aren't those usually for making you pee more?
They do, but in this specific context, they actually have an unexpected effect that helps with water reabsorption.
How does our body actually keep everything perfectly balanced when it comes to acids and bases? I mean, it sounds super complicated.
It is, but the kidneys are like the grandmasters of balance. They play a huge role in maintaining acid-base equilibrium.
The kidneys? Really? I always thought of them just filtering stuff out.
Oh, they do way more. They're constantly working to keep our pH, that's pH, levels stable. It's vital for every single cell.
So if our pH goes off, things go wrong.
Exactly. Too acidic or too alkaline, and our body can't function. And here's where some key players come in.
Like who?
Think of carbonic anhydrase for starters. It's a super important enzyme.
Carbonic anhydrase. Got it. What does that do?
It's central to the carbonic cycle, which is our body's main buffering system. It converts carbon dioxide and water into bicarbonate and protons.
Wait, so it's making acids and bases?
Yeah, it's managing them, making sure they're in the right form at the right time. And the kidneys decide what to do with them.
And what about this proton pump I've heard about?
Ah, the proton pump.
That's how the kidneys actively get rid of excess acid, pushing those protons out into the urine.
So it's literally pumping out acid?
That's a good way to think about it. It's called the vacuolar H plus ATPase.
H plus ATPase. Okay, and what if our body gets too alkaline?
Then the kidneys switch gears. They can actually inhibit the proton pump and start secreting bicarbonate instead to bring the pH down.
No way! So they have an up and a down switch?
Absolutely. It's incredible how adaptable they are. They really are the unsung heroes of acid-base balance.
So if the kidney is this master regulator, how does it actually reabsorb bicarbonate, right? That's key for pH.
That's where carbonic anhydrase comes in. It's an enzyme, super crucial.
Carbonic anhydrase? What does it do exactly?
It basically catalyzes the whole bicarbonate cycle, keeping everything balanced with carbon dioxide and carbonic acid.
So it's like a rapid interchanger?
Exactly. In the kidney tubules, it's constantly converting things.
Okay, so take me through the steps. How does the kidney recover bicarbonate?
All right. First, metabolic acid produces a proton, which gets secreted into the tubular lumen.
So a proton goes into the tube.
Then, this proton combines with filtered bicarbonate to form carbonic acid.
Carbonic acid.
Which then spontaneously breaks down into water and carbon dioxide.
Water and CO2, easy to cross membranes.
Right. The CO2 zips back into the cell.
And once it's in the cell,
carbonic anhydrase converts it back to bicarbonate again, regenerating it.
Wow. So it's essentially recycling bicarbonate back into the plasma.
Yep. Pumping it right back to maintain our systemic buffer.
And the kidney reabsorbs all of it, every single millimolar.
Every single millimolar of the 25 millimolars filtered. It's a bicarbonate recovery machine.
Okay, so that's the bicarbonate cycle. What about the proton pump? How does that work in urinary acidification?
Right, so the kidney also has the special pump, the vacuolar H plus ATPase.
A P what?
ATPase. It's a proton pump. It actively pushes protons into the urine.
Ah, so it's like literally pumping out the bad stuff.
Exactly. It uses ATP to export hydrogen ions into the tubular lumen.
And that acidifies the urine, right?
Yes, precisely. This acidification is crucial for getting rid of metabolic acids.
So it's a full cycle to get those acids out.
It is. First, the pump secretes the hydrogen ion.
Okay, acidifying the lumen.
Then this hydrogen ion combines with bicarbonate, which forms carbonic acid.
Which then breaks down into CO2 and water.
You got it. The CO2 diffuses back into the cell.
Where it's converted back into bicarbonate.
Yes, and that bicarbonate is then transported back into the plasma.
Wow. So it's not just getting rid of acid, it's regenerating bicarbonate for the blood too.
Absolutely. It's a dual-purpose system, eliminating acid and maintaining that systemic buffer.
So beyond just bicarbonate, are there other ways that kidneys handle these protons?
Absolutely. Think of it like a backup crew. Phosphate and sulfate are key players here.
Phosphate and sulfate, how do they jump in?
Well, filtered phosphate, specifically HPO4 2-, grabs onto secreted protons.
And not forms what?
Biphosphate, H2PO4, which then gets peed out. So protons gone.
Got it. And sulfate does something similar.
Exactly. Sulfate, SO4 2-, also captures protons, forming sulfuric acid.
Which also gets eliminated, helping with proton removal?
Yep, another proton gone. But wait, there's a really cool process called ammoniogenesis.
Ammoniogenesis sounds intense.
It's brilliant. The kidney takes glutamine, an amino acid, and converts it into ammonia, NH3.
Okay, ammonia. What happens next?
That ammonia, which is permeable, diffuses into the urine, into the lumen. And there...
it meets a proton.
Yes. NH3 plus H plus equals ammonium, NH4 plus.
So it traps the proton there.
Precisely. Ammonium is charged, so it can't just diffuse back out. It's trapped and excreted.
That's super clever. And this also helps with bicarbonate.
It does. This ammonium trapping indirectly helps reabsorb more bicarbonate too, maintaining that essential plasma buffer.
So after all this amazing buffering, how does the kidney actually adjust if things get too acidic or too alkaline?
That's where the real regulation magic happens. It responds differently to acidosis and alkalosis.
Okay, so like a metabolic balancing act?
Exactly. In alkalosis, when there's too much base, the kidney needs to get rid of bicarbonate.
And how does it do that?
It basically hits the brakes on the proton pump, inhibits it.
So it's not pumping out as many H plus ions?
Right. And it starts secreting bicarbonate directly into the urine to lower blood pH.
Wow, it actively secretes it? That's wild.
It does. But then in acidosis, when there's too much acid, it's the opposite strategy.
So it ramps up the proton pump?
Yes, increases its activity, pushing out more H+.
And what about the other buffers we talked about?
It also boosts ammoniogenesis and the excretion of phosphate and sulfate.
To get rid of those extra protons?
Precisely. It maximizes every system to restore balance.
So it's constantly adjusting based on the body's needs.
A true master of acid-base equilibrium.
How our bodies keep everything balanced, specifically the bicarbonate buffer system.
Yes, this is the main event. It's basically our body's lead bodyguard for blood pH.
A bodyguard, I like that. So what exactly does it guard against?
Against big swings in pH, keeping our blood from becoming too acidic or too alkaline.
And how does it do that? Like, what's the magic trick?
It's all about a reversible chemical reaction. Think of it like a seesaw.
Okay, a seesaw, I'm picturing it.
On one side, you have carbon dioxide and water. They combine to make carbonic acid.
And the other side?
That carbonic acid then splits into bicarbonate ions and hydrogen ions. It's constantly going back and forth.
So if your blood gets too acidic, what happens on the seesaw?
It shifts. The system absorbs those extra acid-making hydrogen ions. It's like the seesaw is balancing everything out.
That's incredible. So it's constantly adjusting in real time.
Precisely. It's our most critical system for maintaining pH homeostasis. Without it, we'd be in serious trouble.
So how exactly does this bicarbonate buffer system work on a chemical level? What are the key reactions?
Okay, so it starts with carbon dioxide, CO2, and water. They combine. This is called hydration.
They just combine naturally?
Yes, but there's a super fast enzyme called carbonic anhydrase that makes it happen almost instantaneously.
Oh, so it's like a catalyst.
Exactly. It forms carbonic acid, H2CO3. This acid is the crucial middle step.
And then what happens to the carbonic acid?
It's unstable. It quickly dissociates or breaks apart into bicarbonate ions, HCO3 minus, and hydrogen ions, H plus.
So CO2 and water turn into carbonic acid, which then splits into bicarbonate and hydrogen ions.
You got it. And this whole process is reversible.
Reversible? What does that mean for our body's pH?
It's everything. If your blood becomes too acidic, meaning too many H plus ions, the bicarbonate ions grab onto them.
To neutralize them?
Yep. They form carbonic acid again, which then turns back into CO2 and water.
And we just breathe out the CO2.
Precisely. If your blood is too alkaline, meaning not enough H+, then the carbonic acid dissociates more, releasing more H+.
So it's like a chemical seesaw, constantly balancing the H plus ions.
A perfect seesaw. That's how it handles both acidosis and alkalosis, keeping your blood pH super stable.
So it's like this chemical dance, right? What about our breathing? How does that play into this whole bicarbonate system?
Oh, the respiratory system is huge. It directly controls CO2 levels in your blood.
Like if I breathe faster.
Exactly. You exhale more CO2.
And CO2 is acidic, so that would raise my pH.
Yes, less CO2 means less carbonic acid, pushing the balance towards a higher pH. It's called respiratory compensation.
So if my body is too acidic from, say, exercise,
your lungs kick in. You start breathing heavier, hyperventilating a bit to blow off that extra CO2.
And that helps bring the pH back up to normal.
That's the goal. It's a rapid response to metabolic acidosis.
What about the other way around, if my blood is too basic?
Then you'd breathe slower, holding on to more CO2, which makes the blood more acidic to bring the pH down.
So the lungs are like a fast-acting pH balancing fan.
A perfect analogy, pushing out or holding on to CO2 to tweak the blood's acidity or alkalinity.
That's incredible. The body just knows to do that. So it's clear the bicarbonate system is a superstar, but are there other players on the field?
Absolutely. It's not the only one. The body has a whole team of buffers.
Like who?
Well, there are protein buffers, phosphate buffers, and even hemoglobin buffers.
Oh, so not just bicarbonate running the show.
Exactly. The bicarbonate system is the fastest in plasma, super immediate.
Super immediate. We're talking seconds?
Yup, seconds. Its capacity is high too, mostly in the blood.
And the others, what do they do?
Protein buffers like albumin work in minutes to hours. They have moderate capacity both inside and outside cells.
So they're a bit slower then.
Yes, but they're important. Then you have phosphate buffers, which are slower still, hours.
Hours, wow.
They're mostly inside cells, and their capacity is a bit lower.
Okay, and hemoglobin?
Hemoglobin's a big one too. It binds to hydrogen ions, primarily in red blood cells. Very high capacity, and it kicks in fast, seconds to minutes.
So different speeds, different strengths.
Precisely. Each buffer has its role, working together for pH balance.
A true team effort then, not just a one-man show with bicarbonate.
That's right. They all contribute to keeping the body's pH just right.
So after all this amazing science, how does understanding the bicarbonate buffer system actually help in the real world?
That's the best part. It's super practical, especially in medicine.
Like, for doctors?
Exactly. They constantly measure blood pH. A normal value is around 7.4.
And if it's off?
If it deviates, it tells them something's wrong, and the buffer system is trying its best to compensate.
So it's like a warning signal.
Totally. And sometimes they need to step in, like with exogenous bicarbonates.
Exogenous? What are those?
Think of it as giving the body extra bicarbonate, often through an IV, to help fight severe acidosis.
Like a jumpstart for the buffer?
Yes, precisely. It leverages the system's buffering capacity to quickly bring the pH back up.
And how do they know if it's a breathing problem or a metabolic one?
Great question. Clinicians look at the pH, PCO2, and bicarbonate levels.
PCO2 being the carbon dioxide?
Right. If the PCO2 is off, it's likely respiratory. If the bicarbonate is the main culprit, it's metabolic.
Oh, that makes sense. So they can pinpoint the issue.
Yep, it's like detective work. Analyzing those three values helps them identify the root cause of the imbalance.
And then they can treat it properly.
Exactly. This system is crucial for guiding clinical decisions and saving lives.
Wow, what a journey through the renal system. It feels like we've covered so much from the ground up.
Absolutely. We started right at the basics with the anatomy, building that foundational understanding.
And we zoomed out to the big picture of overall renal physiology.
Exactly. Understanding how it all fits together, the kidney's main roles.
Which is huge, considering how complex it is. So many functions.
Totally. Then we dove deep into the nephron itself, the real workhorse, right?
The functional unit, yes, all the tiny filtering and reabsorbing happening there.
And that's where we really broke down the intricate processes.
Like how it maintains balance? Because we also covered major metabolic imbalances.
Oh, for sure. The acid-base balance, electrolytes, the things that can go wrong when the kidneys aren't happy.
Right, because the kidneys are like the body's super sophisticated filter and regulator.
A perfect analogy. They manage so much more than just waste.
So basically, we started with where everything is, then what it does, then the specific little machines doing it.
And what happens when those little machines hit a snag and throw everything off.
That's a pretty comprehensive journey.
I think so. A solid foundation for everything to come.
Which is exciting because it means we're ready for what's next.
So it sounds like this whole series really aimed to simplify some super complex stuff.
Exactly. My main goal was always clarity.
Clarity is key, especially in medicine. You want to cut through the jargon, right?
Absolutely. And not just clarity, but sparking that intellectual curiosity too.
Hell, for sure. You want people to ask why and not just memorize.
Exactly. That's how true understanding happens.
So for anyone listening who found this super helpful, what's the best way for them to connect?
Please, just leave a comment. Tell us what you thought.
Comments are awesome. Good, bad, questions.
All of it. We love hearing from you.
And to stay up to date, like with new episodes coming out.
Follow us and give us a like. That's the best way to not miss anything.
So like smash that follow button, give it a big thumbs up.
Precisely. It helps us know what resonates.
It really creates a community too, right? Everyone learning together.
That's the dream, building that intellectual hub.
Because medical education can sometimes feel a bit isolating.
It can. So we want this to be a shared journey.
Love that. So comments, follows, likes, all the good stuff.
All the interaction helps us grow.
So that wraps up our deep dive into all things renal physiology. But what's next for us?
Oh, this is where it gets really exciting. We're moving from the how it works to the what goes wrong.
What goes wrong meaning?
Nephrological diseases, the main ones.
Whoa, that's a big jump. How are we tackling that?
With the clinical approach, we'll be looking at diagnostic reasoning.
Like thinking like a doctor?
Exactly. How do you put all those pieces together when a patient walks in?
So it's not just memorizing diseases?
Absolutely not. We're integrating everything we've learned.
Physiology, pathophysiology.
And clinical medicine, all into one cohesive framework.
That sounds incredibly practical.
It is. It's about seeing the whole picture, not just isolated parts.
So we're going from understanding the individual gears to seeing how the entire engine breaks down and how to fix it.
Perfect analogy, from the foundation to troubleshooting.
I'm really excited for that. It feels like the next natural step.
It is. It builds on everything we've built, making it applicable to real-world scenarios.
Wow, so that's a perfect lead-in to wrap things up here, isn't it?
It really is. We've covered so much, from the foundational physiology to where we're headed next.
And what an incredible journey it's been through the renal system.
Absolutely. I hope everyone feels as excited as we do about understanding it all.
We really want to thank our amazing listeners for sticking with us through this whole series.
Your curiosity and engagement are why we do this.
Exactly. So a huge thank you from both of us.
And please don't forget to follow us, leave a like,
and especially comment. We love hearing your thoughts and questions.
It helps us tailor future content and makes this a true conversation.
So with that, we'll see you in the next episode for the deep dive into nephrological diseases.
Get ready for a whole new level of clinical reasoning.
It's going to be fascinating. Thanks again for tuning in to the Clinical Medicine Podcast.
Bye for now, everyone.
See you soon.
All right, I think that covers it.
Yeah, definitely.